Okay, so one way that it can be kind of fun to try to learn some of these repair pathways a little bit, really is just to think a little bit more about them and try to understand them better. It's actually think about how expensive each type of repair pathway is. And it's kind of a fun exercise to go through what those costs of doing this repair are for the cell, because sometimes it can be a little surprising, but it also forces us to think through the details of the repair process. And again, for me, I find that this can be a little bit helpful for just trying to learn the things. So we're going to kind of go through a bill, the cost of doing the different types of repairs, whether it's alkylation, mismatch repairs, basic sites, UV dimers, so we can think a little bit about how much energy the cell has to spend to actually maintain this genomic integrity. And of course, what we will discover very quickly is that it is not cheap, it is not free to do these types of repair processes. And which ones are the most expensive may actually surprise you a little bit. So let's go through each one and think about the costs that are associated. So let's go through the, let's think about direct repair for a second, okay? So here we're thinking about, we've got some alkylated base. So let's just draw one. Let's make it guanine for fun. So let's draw this out. It's also a great time to review your drawing skills, your nucleotides. So a guanine would look like this. Okay, and if this were to be alkylated, sorry, let me finish that. And if this were to be alkylated, it would look, let's put it on the O6 position here. So now we've got, say, a methyl group here. Now we've messed that up. Okay, so we've got this alkylation here. And, oops, let me just finish drawing. I always forget the 5' end. But you've got it here. And we talked about how that, that methyl group that's on the O here can be removed by an enzyme called AGT. So AGT has this cysteine residue on it, can come and deal with this. Pick up an H. And in this way, restore it can restore our AGT, right? Or sorry, restore our base. So we've dealkylated the guanosine back to regular guanosine, and in the process we've transferred this methyl group here. And at first blush, this may not look like it's very expensive, because all that was needed was for this methyl group to get transferred to this protein, this methyl transferase, and typically we would think that as an enzyme there'd be some mechanism which would eventually regenerate our AGT so that we can do this again. However, this is typically not the case. And in fact, it is almost always the case that the methyl transferase is permanently modified when it recovers this alkyl group from the nucleobase. And so as a consequence, every time we remove one alkyl nucleobase, from our nucleobase, it costs us an entire AGT protein. And this protein is approximately 200 amino acids long. And as you will learn later on, the cost of making this protein is going to be at least 600 ATP, if not more. Okay? And so to do this one type of repair, this direct repair, is actually going to cost a huge amount of energy because it requires us to make a new AGT protein every single time that we do this. Okay? So direct repair, while looking fiendishly simple, is actually quite expensive. Now, how about the cost of something like mismatch repair? How much does this cost the cell? Okay, well, remember that mismatch repair works where we've got our parental strand that has some methyl groups on it, and we've got some daughter strand for which there's a mismatch, usually denoted in this way here. And in order to repair it, what's going to happen is that we're going to assemble a complex here of proteins that are going to basically spool. They're going to recognize this mismatch, and then they're going to spool the DNA around until they encounter one of these methyl groups on the parental strand. And at that point, they're going to cleave at that location. And this will result now in the daughter strand being nicked in this location. Whoops. So we've got a nick here at the site where the methyl group was. Okay. And then the complex is going to chew back with an exonuclease activity until it gets to the mismatch. So now we are left with something like this here, where we've removed the mismatch. Okay, and we have this big empty stretch here. And then this is the point at which the polymerase and ligase can come back in and fill this in and repair here. And so with this type of repair pathway, one of the big things is the spacing between methyl sites sets the fragment size that you need to make, right? So if your methyl groups were very close together, then this fragment might be small. But if your average spacing between methyl groups is quite large, then the distance between the mismatch and the methyl group that is going to set the boundaries of this thing can be quite large. And oftentimes, the spacing of the methyl sites can be on the order of 1,000 nucleotides or more. That's not to say it's always the case, because you know, they're distributed non-uniformly throughout the genome, but in some cases the nearest methylation site might be 1,000 nucleotides or more. And so if you think about how many DNTPs that would be, so like 1,000 DNTPs at least to repair something that's 1,000 nucleotides here. So that turns out to be pretty expensive. And that's a lot of fuel to burn to fix a single, remember this is a single mistake here, okay? And so this could be a huge amount of energy to fix a single mistake here. You know, one way around that would be to put more methyl sites throughout the genome to reduce the average spacing here, but it also costs energy to deposit these methyl groups as well. So, you know, it's sort of a cost-benefit analysis that kind of goes into working this out. But so mismatch repair can, yeah, be surprisingly expensive. Okay, so how about base excision repair? How much is this going to cost us? So remember, we're thinking about a scenario here where, say, we had some sequence A, T, C, G, T, like this, and it had originally been paired T, A, G, C, A, like you see here. Okay, and then through some deamination process here, something might happen to where that C now becomes, say, a U. So uracil would be what you get from cytosine deamination. And so now, in our two strands here, uh-oh, not good at all, right? We've got this problem here. And so with base excision repair, the idea, recall, is that we're going to recognize this, and we're going to cleave the base, right? And that's going to give us something that looks more like this now. AT blank GT. Okay, and then over here, T A G C A. Okay, so now we have this A basic site here, which is, you know, kind of like a sad empty space. And that's going to get recognized by AP nuclease. Oops. It's going to get recognized by AP nuclease, so we'll get an AT, and we've got this little flap hanging over here with a 5' phosphate, and we've got the G and the T here. T A G C A. So we've got all of those. And the key thing to recognize is that we have a 3' OH here. We've got base pairs here. And then we've got a single nucleotide here of single-stranded DNA, whereas this stretch here is double-stranded DNA. And from here, it's simply a matter of polymerase using the 3' OH here to go ahead and extend, and it will put a C here. And as I mentioned, depending on the mechanism that's used, it can continue to make a few extra nucleotides, so maybe it makes an extra 5 to 10 nucleotides, or in some cases, a separate pathway will be used in which you cleave off the deoxyribose phosphate here at the end, and then you stitch it together with ligase. But regardless of which path you choose, for the base excision repair, basically we needed only to cleave the base, cleave the backbone, and then do a few short additions of nucleotide followed by ligation to stitch it together. And so with this, you might be looking at a repair cost of anywhere between, you know, 2 to 10 ATP, something like that, to fix this. And of course, that will depend a little bit on whether you do this extension part up here. So in contrast to mismatch repair and in contrast to mismatch repair and direct repair, this is a much more cost-effective solution to fixing these base problems. Okay, so one last scenario to consider, and that is nucleotide excision repair. Okay, and so remember, this is the pathway that repairs things like pyrimidine dimers or larger damaged stretches like that. And so let's assume we've got our parental strand and we've got our, we've got two strands rather, not parental and daughter. We've just got two strands. And we've got some of these UV dimers in this region here. So now we've got like this bulged structure here that's come from those things being connected together here. And with nucleotide excision repair, what's going to happen is that we are going to assemble a complex here that's going to recognize the strand that contains the thymine or pyrimidine dimers, and then it is going to cleave the strand that it has that has those dimers on both sides, and this will generate a nicked substrate like you see here. Which got these still connected, and of course we have our thymine dimers and some stuff here. And this fragment is typically approximately 13 nucleotides long. And once these nicks have been made, we can use a helicase activity here to separate the strands. So we have now this side, and we have the two halves there, and then we've got displaced now is that 13 nucleotides that has that dimer in there, you see here. And then once we have done that, then it's as simple as all the other pathways we've looked at before, in which the gap can be filled in by extension here. So if this was the 5', 3', 5', 3' ends here, then it's a simple matter to extend for polymerase to come in, fill this in. So polymerase plus ligase will fix this problem, just like we've seen many times before. And so if we do our bookkeeping again, you know, this because this nucleotide excision surveillance complex up here usually recognizes these structures and then cleaves to generate a fragment that's about 13 nucleotides long, then we know there's going to be about 13 DNTPs needed for this. The helicase activity is going to require some energy, so some nucleotides are going to be burned there. And the ligase activity will also need ATP. So you're looking at somewhere between, you know, 20 to 40 ATP to characterize or to fix this mistake. Now these aren't numbers that you need to memorize. They're just kind of back of the envelope calculations that come from looking at what's going on here. And so if we were now just to kind of like rank the costs of the damages, what we found from our analysis was that mismatch repair could easily be more than 1,000 ATPs worth of cost, we found that direct repair, because an entire protein is consumed in the process, this could be on the order of 200 ATP or more to fix these very simple alkylation problems. Then we found that nucleotide excision repair was on the order of 20 to 40 ATP. And then finally, base excision repair could be, less than 10 ATP in some cases. Really, so theoretically, could get down to 2 or 3. These costs here, and so we can see that fixing the different problems that occur to maintain genomic integrity, it always is going to cost something, and in some cases, some mistakes are much more expensive to fix than others. And it's worth kind of noting that with things like mismatch repair, some of this cost comes from the fact that you have to have a way of telling daughter and parental strand apart in order for the repair to work. So you have to invest energy in that methylation strategy, and it's really critical that you find a methylated nucleotide in order to tell you which strand is which. And so that's what can rack up these expensive ATP bills here. With the direct repair, the cost really comes from the sacrificial nature of the repair process. So if we didn't have to destroy the protein every time we did a repair, this could potentially be a lot cheaper. But as it is, each repair requires an entire protein to be made, and that's looking at least 200 ATP, possibly even higher. It could be as high as 600. It's really just an expensive process to make those proteins, depending on their size. And then you can contrast these two processes here to the expenses of the two more surgical repair systems. So nucleotide excision repair, since you're always removing a roughly 13 nucleotide stretch, your costs are nowhere near as expensive as they are with these. And with base excision repair, since you're really restricting your repair locations to the exact damaged base, these can cost as few as a couple bases. So even though sometimes it seems simpler, the solution of direct repair seems very simple, and we might think it's not so expensive compared to some of these complex repair systems. From an energetics perspective, they kind of rank out like this. And as I mentioned, this isn't something you need to necessarily know for the course. It's just a fun way, I think, of trying to learn a little bit more about each repair pathway, makes you think about what's going on, and then helps you kind of internalize what's going on with each one. So I hope that was helpful, and I'll see you in the next video.